Hello everyone and welcome back to C.A.R. Entertainment. I am your host Dylan Hamilton, guys, and today we're going to be moving on from where we last left off in HU1 biology, which was neurons. We left off talking about action potentials and how it may start to get moved across from a resting potential to an action potential. And so now we're going to go in depth with this here in the nerve impulse. We're going to talk to you all the way through it and how saltatory conduction and all works. So let's go straight in depth. Well, so the resting potential then. So the resting potential is including so neurons have this thing called a potential difference, right? And this happens across the membrane. Neurons will become polarized as an electrochemical gradient across the membrane. This is caused by excess positively charged ions on the outside compared with the inside. We talked about this last time, saying that there's basically more ions on the outside of the axon than the inside of the axon. And when this is the case, then the axon will have a resting potential. This differential is maintained as the membrane is impermeable to the flow of ions when not conducting an impulse. Therefore, ions cannot come from the outside to the inside as the membrane is impermeable to it. So an action potential then. How does an action potential actually occur? Well, when the neuron is stimulated, the membrane will become permeable to ions. Okay? So basically, whenever it becomes stimulated from your dendrite and your cell body, etc., then it will become permeable to ions. An excess of positive ions on the outside, so as we know there's more ions on the outside to the inside, these will then diffuse from the outside into the inside down a concentration gradient. The potential difference will decrease as gated ion channels will open, rapidly increasing the rate of diffusion, and this will lead to depolarization of the neuron, meaning that the inside is more positive relative to the outside. Both are still positive, but the inside is more positive, and this is the depolarization of the neuron. So depolarization is called an action potential, and the sequence of events is roughly about 1 millisecond. The peak of the action potential, whenever you reach the peak of the action potential, then the recovery phase starts. And this is where positive ions will diffuse and be pumped out of the neuron to the outside again. This will rapidly restore the resting potential and the membrane will become impermeable again. Now during this recovery phase, further impulses will not occur as the gated ion channels will be closed, and the entire electrochemical sequence is 4 milliseconds. So as we see here in the diagram, you have your threshold of excitation and moves up, and at that point then you go straight to your action potential, and then repolarization, this hyperrepolarization we'll talk about now. So the changes in potential difference across the axon membrane as an action potential. So the resting potential will start off inside the axon at negative 70 millivolts relative to the outside. It's still positive, but it's just 70 less than the outside. The membrane will become permeable and positive ions will diffuse into the axon, making it depolarized. It will then be negative 40 millivolts as gated ion channels will open and positive ions will flood in at a rapid rate. So basically, once the gated channels open, the inside is then 40 millivolts less than the outside. And then whenever these ions come in, rapid depolarization will take place, meaning the inside is positive 40 millivolts to the outside, meaning it's 40 more. And this is the action potential occurring. As positive ions diffuse out and are pumped out of the axon, the refractory period will occur and the membrane will not be depolarized again until the resting potential is restored. There will be a slight overshoot as inside the axon is slightly more negative, and this is called hyperpolarization. The resting potential will be restored and the axon will then be able to conduct another impulse if it is stimulated. So the refractory period, and this refractory period is known as repolarization in the diagram there, and the functions of this is that it ensures action potentials are propagated in one direction only. This will limit the number of action potentials and ensure each action potential is a discrete entity. So the features of the nervous system, there's two very important features. One is threshold stimulus, which is the level of stimulus the neuron requires before an action potential is produced. So basically, how much needs to occur before the axon can actually generate an action potential. Then there's the all or nothing law, which is a principle that once the threshold stimulus has been reached, an action potential will result, and different intensities of action potential do not occur. Once an action potential occurs, it will sweep along the neuron as a nerve impulse. So the propagation then, how does it move along the axon? Well, action potentials move rapidly as a wave of depolarization moves quickly. Now, you have a region in the neuron where the action potential will take place, and the region immediately behind this depolarized zone of an action potential is repolarized. So ions will move from the outside from the inside to the outside immediately behind the depolarized zone. This one part that is depolarized will set up local circuits, and these areas will be immediately adjacent on either side. so basically forward and back and on the outsides. The positive ions in the depolarized zone will pass along the inside towards the polarized zone, and on the outside, positive ions will move back from the still polarized zone into the depolarized zone. Now, this can be quite complicated, so the best way to do it is basically just draw out a diagram and then put like 3 different rectangles. The middle one will be the depolarized zone, and then the one behind it and the one in front will be your polarized zones. And basically, ions will move from different places. So we'll see positive ions in the middle rectangle will move along the inside towards the polarized zone, so they'll move forward to the right, and then the outside you'll have ions on the outside which will move back from the still polarized zone into the depolarized zone. So that'll be from the right one into the depolarized zone in the middle, and then that'll also work for the one behind it as well. Very complicated, I do know. I would recommend Googling it if you still don't understand it, and maybe get a diagram on it. Processes occurring continuously will create a wave, and depolarization will move rapidly. Similar circuits will enable a resting potential, which are restored directly behind an action potential. There are different factors that affect the speed of a nerve impulse, such as the presence or absence of the myelin sheath, as an electrical insulator preventing depolarization. So whenever the myelin sheath is on the axon, an action potential cannot occur, and this is why nodes around the are present, because they allow depolarization to occur. These local circuits will form between these nodes, and so the section of the neuron will be bypassed, meaning that everything that's covered by a myelin sheath will be bypassed, and it will just jump from node around the node to around the node to around the. So as we see, antennas will jump from one node to the next, and this is called saltatory conduction. You also have the thickness of the axon, which is whenever the diameter is larger and thicker, therefore it will have a faster impulse as it's proportionally less leakage of ions. If there's too much leakage, then it's difficult to maintain potential gradients which are required from the resting and action potentials. Myelinated neurons also have fewer ion channels under the fatty myelin sheath, and so myelination can overcome problems of a small diameter. Furthermore, temperature will affect the rate of diffusion as it affects the speed neurons can conduct impulses. Thanks for watching this video of the nerve impulse. I'll see you next time. I think it's for synapses we're going to be doing. I've been Dylan Hamilton from C.A.R. Entertainment, and I will see you next time.